Proteins that bind along the length of microtubules are collectively called microtubule-associated proteins, or MAPs for abbreviation. These proteins not only influence the microtubule dynamics, but also they're important in organizing microtubules and also facilitating interaction of microtubules with other cellular components. So let's take a look at these protein molecules. MAPs play especially a very important role in neuronal biology, where they stabilize microtubule bundles from the core of the axon and dendrites that extend from the cell body. Here in the micrograph, you can see a neuron. It is a hippocampal neuron. You see two colors in this micrograph. One is orangish yellow color, other is green color. The green color is basically the tau staining. This type of map is especially localized on the surface of microtubules in axons. The orangish yellow color is the MAP2 staining. This staining, as you can see, is restricted pretty much in the cell body and the dendrites. Basically, because of these maps, microtubules in axons and dendrites have different arrangements and they interact with different cellular components. Neuronal maps have at least one domain that binds to the microtubule surface and another that projects outwards. The length of projecting domain basically determines how closely map-coated microtubules can pack together. So if the length of the map is relatively large or longer, the microtubules, when they are arranged, they will be arranged further apart from each other, versus if the length of the map was shorter, then microtubules can be packed closer together. I'll show you arrangement of microtubules in a cross-section in the later slide. But here I would like to mention that microtubules in cells overexpressing MAP2 are widely spaced because MAP2 has a long projecting domain. Here in this diagram, for example, this is a microtubule, and you can see MAP2 has a longer domain which is projecting outwards, and it has a domain that is binding the microtubule. This domain that is projecting outward, this MAP2 domain, is longer than this domain of tau. Also notice that tau is binding, has two domains that can bind microtubule lattice. Both its amino terminus and the carboxy terminus contains these domains. Here is a cross section. You can see that the arrangement of microtubules, these dots are basically cross sections to microtubules. These are basically the cross sections, and you will just see when you look at a cross section of a microtubule in a cell, it will appear as a dot. So things that you would notice here is microtubules are evenly spaced. This is because of the domain of the map that is projecting out from the microtubule. It is a constant length, so microtubules are evenly spaced. These microtubules are coated with MAP2. They're further apart compared to the microtubules in this panel. The microtubules in this panel are basically taken from a cell which was overexpressing tau. Look at the even distribution, and also the point here with this micrograph is the microtubules are relatively closer to each other in the cells expressing tau versus the cell that is expressing MAP2. Microtubule binding domain of several MAPs include multiple copies of tubulin-binding motif. When MAPs are added to a solution of pure unpolymerized tubulin, they greatly accelerate a nucleation process because they can stabilize these small tubulin oligomers that we saw in one of our videos. MAPs are the target of several protein kinases because MAPs too have to be regulated. MAP phosphorylation can control MAP activity and localization. There is an ubiquitous MAP called XMAP215, has close homologs in organisms that range from yeast to human. XMAP basically stands for xenopus, which is frog, microtubule associated proteins, and the number refers to its molecular weight. XMAP215 stabilizes free microtubule ends and inhibit their switch from growing to shrinking state. During mitosis, XMAP215 phosphorylation inhibits its activity because, as you know, during mitosis, cells get rid of the microtubule array of the interface. Microtubules basically depolymerize, centrosomes duplicate, they move to the opposite poles, and then microtubules again start coming off the centrosome through that ring complex made by the gamma tubulin. This phosphorylation of XMAP215 inhibits its activity, and as a result, the dynamic instability of microtubule increases tenfolds. So MAPs play a very important role in organizing microtubules, influencing their dynamic instability, and also they promote or inhibit their interaction with the rest of the cellular machinery.